So hello dear students, today I am going to tell you regarding the mechanism of action of the fluoroquinolones. Okay, mechanism of action of fluoroquinolones. So let me tell you that fluoroquinolone is mainly active against the gram-negative bacteria, mainly, and it can having the spectrum to affect the gram-positive bacteria also. Now, before understanding the mechanism action, how fluoroquinolones acts on the bacteria, let me tell you first because fluoroquinolones acts on the replication side of the bacteria. So to understand the mechanism of action of fluoroquinolones, you need to understand how the bacteria divides. You know, mainly the bacteria divides. Say for example, this is your bacteria, and it's having circular DNA. Okay, this is the circular DNA. Okay, let me make it circular DNA of the same color. This is the circular DNA. You know, the bacteria divides by binary fission. Binary fission, in which this cell produces two daughter cells, like this way, 1, 2, and each daughter cell is having a part of circular DNA, parent circular DNA, and a new complementary strain. This is known as semi-conservative method because it's having one parent DNA and another is the complementary DNA. That is, after replication, the DNA form. Now, what is going to happen here? Say for example, I will take this part of the DNA. You know, the DNA is double helical in structure. Okay, so this is your DNA. Okay, enzymes come into the action. First comes helicases, DNA helicases. Say this is DNA helicase. What is the function of the DNA helicase? It introduces a nick. It introduces, say for example, this is your DNA helicase, and it introduces a nick. And when it introduces a nick, your DNA looks like this way. See, when it introduces a nick, there occurs separation of the DNA. This is called nuclear replication fork. Again, the replication fork, then your new enzyme come into action. This is your DNA helicases, helicase. Then after that, DNA polymerases come into the action. DNA polymerases read the codon and forms the complementary strain. It is DNA polymerase. DNA polymerase. Okay, it forms the complementary strain. Now, see what is going to happen here. DNA helicases introduces a nick, forms a replication fork. But when it forms a replication fork, see, it introduces a positive supercoiling at both sides. Here, your coil become positively coiled each other. Positively supercoiled means positive supercoiling is introduced by the nick of this DNA. So, in order to move further, this supercoiling must gets removed. Otherwise, there occur no separation. For the removal of this positive supercoiling, there is one more enzyme that is known as, this is the enzyme. This is known as DNA gyrase or topoisomerase 2. You know what is topoisomerase 2? It is like this enzyme, like this way. It's having two hands, topoisomerase 2. One end is having cutter and another here is having ligase activity. It seals the thing. This DNA helicase, what it introduced, it cuts down the one of the strand with the help of this cutter. It cuts the one strand of DNA, and when it gets relaxed, when you cut that one strand, it gets relaxed. After relaxation, then the DNA ligase seal the things. Okay, seal the things. Now you are getting your strand like this way. One is the parent strand, another is your complementary strand.